Not everything that tastes bitter is potentially harmful. This might seem counterintuitive, but a recent study provides an explanation. While bitter taste is traditionally a warning sign of toxicity, some bitter substances are actually nutritious and vital for humans. Let's delve into the fascinating findings of this study and understand why not all bitter tastes are bad. Our sense of taste is crucial for making food choices. Among the 5 basic tastes, sweet and umami indicate energy-rich and nutritious foods. Salt helps maintain electrolyte balance, while sour flavors warn us of unripe or spoiled food. Bitter tastes, on the other hand, have long been associated with potentially toxic substances. This makes sense given the presence of toxic plant substances like strychnine and hydrogen cyanide. It's also plausible that babies and toddlers reject bitter foods, as even small amounts of toxic substances can be harmful to them. However, not all bitter substances are dangerous. Some peptides and free amino acids, which taste bitter, are non-toxic and nutritious. This paradoxical phenomenon has puzzled scientists for years, but a new study offers some answers. An interdisciplinary research team led by molecular biologist Mike Behrens at the Leibniz Institute for Food Systems Biology at the Technical University of Munich has investigated this phenomenon. Using an established cellular test system, the team discovered that 5 of the approximately 25 human bitter taste receptor types react to free amino acids and peptides, as well as to bile acids. These amino acids and peptides are produced during protein breakdown and are abundant in fermented foods like cream cheese and protein shakes. Interestingly, bile acids, which play virtually no role as a food component, fulfill their own functions in the body. They could be considered activators of endogenous bitter receptors located on intestinal and blood cells. This discovery suggests that bitter taste receptors might have roles beyond just warning against toxic substances. They might be involved in important physiological processes. Bioinformatician Antonella Di Pizio explains, "Interestingly, our modeling experiments show that a certain bitter-tasting peptide can adopt a functionally active 3-dimensional shape, similar to that of bile acids inside the receptor binding pocket. This coincidental similarity could explain why the same set of bitter taste receptors react to both groups of substances." First author Sylvia Schaefer adds, "Our genetic analyses show that the ability to recognize both bile acids and peptides is highly conserved in 3 of the bitter taste receptor types and can be traced back to amphibians. This indicates that at least the recognition of one of the 2 substance groups is important across species." Principal investigator Mike Behrens further explains, "Bile acids and bitter taste receptors existed millions of years before the typical bitter substances of today's flowering plants and long before humans, in fish for example. These findings provide new insights into the complex systems of taste perception. They suggest that bitter receptors play additional, as yet unknown roles in human health that go beyond their function in food selection. This research opens up new avenues for understanding the diverse functions of bitter taste receptors and their impact on our well-being."